Thank you so much. So good morning, everyone. And in the next minutes, I will show you some results related to the effect of nitrate supplementation in age-related neuromuscular dysfunction. So neuromuscular aging is characterized by alteration at the level of the motor units, impairments in the denervation-reinnervation factor with failure in reinnervation, which causes a permanent detachment of the neuron from the muscle fibers with observable instability at the level of the neuromuscular junction. Moreover, denervation is correlated with muscle mass loss, and in general, aging is well associated with an increase in oxidative stress caused by reactive oxygen species damage. Nitric oxide is a very small but such important signaling molecule involved in different mechanisms such as vasodilation, mitochondrial respiration and biogenesis, and even glucose uptake. And in muscle, an in vitro study demonstrated its ability to enhance protein synthesis on C2, C12 cells. So nitric oxide can be synthesized, can be produced by two different pathways. So by the metabolism of the exogenous dietary nitrate intake or by the endogenous activity of nitric oxide synthesis. But during aging, one or both pathways can be compromised, decreasing a reduced nitric oxide bioavailability with potential impact on nitric oxide targeted phenomena. So with all these notions in mind, we ask ourselves whether the exogenous nitrate supplementation can induce a beneficial remodeling at the level of the neuromuscular junction and thus counteract neuromuscular alteration during aging. In order to do so, we used wild-type male mice undergoing physiological aging, and we firstly compared those with a group of young mice in order to evaluate the correct time window of aging we wanted to study, 24 months. And then a group of old mice were treated for 2 months with 1.5 millimolar of inorganic nitrate dissolved in drinking water, and at 24 months we sacrificed all the old mice and we dissected gastrocnemius for morphological and molecular evaluation. So firstly, we demonstrate an impaired activity of the endogenous nitric oxide synthase, which has a significantly reduction in its activating phosphorylation levels. And then we even measure the concentration of nitric oxide-based compounds within the muscle, which are significantly augmented following the 2 months treatment, meaning that our treatment has significantly reached the district of our interest, meaning the muscle. So first we start with the evaluation of the cross-sectional area in the gastrocnemius, and we saw a significant reduction of the muscle mass in old mice compared to the young control, and a positive recovery, significant recovery of the mass following nitrate supplementation. And because the muscle mass is controlled by a balanced equilibrium between protein synthesis and protein degradation, we next move on with the analysis of key factors of both pathways. So we explained the muscle atrophy we saw in old mice through the significant overexpression of the muscle-specific atrogen, MURF1 and atrogen, that is confirmed by significant higher level of the polyubiquitinated proteins, which are the ones undergoing degradation, while nitrate supplementation significantly augment the activating phosphorylation of the downstream factor of the mTOR pathway, so S6 and its kinase, demonstrating an in vivo nitric oxide augmented anabolic response in 24-month-old mice. Next, we focus on neuromuscular parameters. So we firstly analyze the expression level of the so-called denervation-related factors. And as you can see here, all these factors are strongly induced in old mice compared to the young control. And nitrate did not modulate their expression level, but when we focus on the expression level of NCAM1, which is a very important adhesion molecule that is overexpressed following denervation in an attempt to recall back the motor-neuron interaction, its overexpression during aging show a tendency to reduction following nitrate supplementation, and this data is strengthened by counting the fibers positive to NCAM1, which shows a significant reduction reduction in their percentage following 2 months nitrate supplementation when compared to the old mice, meaning that at least nitrate supplementation is able to modulate the NCAM1 expression level in old mice. Next, we analyzed the neuromuscular junction morphological feature of images acquired at the confocal microscopy. So this panel is quite representative of the data I'm showing you here. And the old mice here it's a quite extreme example, but it's very representative of the highest, of the very high index of fragmentation we saw in old mice compared to the young control. And when we supplemented the mice with nitrate, we saw a significant reduction of fragmentation of the postsynaptic side. And this data is reflected by the percentage of overlap between the pre and postsynaptic sides, in which even if in this graph significances are missing, then we can see how nitrate supplementation has a tendency to ameliorate the overlap between the two sides. And as we know, the maintenance of the structure is activity dependent. So ameliorating an amelioration in the superimposition in the contact between these two sides can reduce the probability to lose this functional and structural organization. So we can speculate that nitrate supplementation can promote a positive remodeling of the neuromuscular junction in old mice. And last, because of the observation related to the junction, we even evaluate the levels of PGC1 alpha, which is a key factor for the transcription of synaptic genes. But we didn't see any change in neither transcript nor protein level of this factor. But we surprisingly saw a significant induction of their up and downstream effectors, meaning that nitrate is able to positively modulate this signaling pathway. And because PGC1 alpha and TFAM are directly involved in the control of the oxidative stress, which is an event that is highly represented during aging, we even evaluate few parameters indicative of the redox status of our mice. So we firstly measured the mitochondrial hydrogen peroxide emission in our mice, and we saw a significant reduction of this emission in treated mice compared to the old control mice. And we next analyzed the levels of carbonylated proteins that again show a reduced significantly reduced levels of carbonylation in treated mice compared to the old control mice. And these values are going down almost at the same level of the young control, so this is a quite positive effect. And last, in order to explain, to find the reason to this trend, we even evaluate the expression level of few conventional antioxidant enzymes. So we evaluate SOD1 and catalase that did not change in any of the conditions under study. So we can say that nitrate supplementation positively modulates the redox status in old mice, but acting through non-conventional patterns. So to conclude, following exogenous nitrate supplementation, we have seen a recovery of muscle mass driven by a boosted synthetic pathway and an ameliorated neuromuscular junction stability. And according to literature, an increased synthetic pathway that in our case is driven by nitrate supplementation can have a possible role in the positive remodeling of the neuromuscular junction as described in this review. And lastly, nitrate supplementation is even involved in the positive control of the redox status in old mice. Last but not least, I would like to thank all the collaborators that worked in this project, all my lab mates, and a special thanks to my supervisor, Professor Pellegrino, Professor Bottinelli, and Professor Brocca for all the scientific support, and you all for your attention. Thanks.